Hi everyone, welcome to the fourth video of the series Biological Basis of Life. My name is Akhilanand Singh. In this video, we are going to discuss gene mutation. The series Biological Basis of Life is dedicated to the Unit 1.7 of the UPSC Anthropology Optional Paper 1. The main objective of the series is to discuss the essentials of biology which are required to understand the biological anthropology. In the first video of the series, we learnt about cells and cell organelles. The second video of the series was about protein synthesis. We talked about DNA replication and cell division in the third video, and this fourth video is about gene mutation. Almost all the examples in this video would be related to humans as we are learning these topics from the viewpoint of anthropology. What are mutations? Mutations mean change. Mutations are changes in the genetic material in our bodies. These are random changes in the nitrogenous bases of our DNA and RNA. You may recollect that our DNA has 4 nitrogenous bases: cytosine, guanine, adenine and thymine. RNA also has 4 nitrogenous bases in which cytosine, guanine and adenine are the same bases which are present in DNA. Only the 4th base is different, which is uracil in place of thymine. Most people have dozens or even hundreds of mutations. Mutations have played a key role in our evolution. Mutations ensure that there are genetic variations amongst all the human beings. Mutations can be neutral, harmful or beneficial. Majority of mutations have no positive or negative impact on us. They do not hamper the protein synthesis process. Many mutations get auto-corrected by the repair mechanism built within our cells. All such mutations are called neutral. Harmful mutations. Some mutations interrupt the protein synthesis process or they completely halt it. These mutations are potentially carried over generations and they may cause cancer. Such mutations are called harmful mutations. Beneficial mutations. Some mutations may provide us strong immunity. They may strengthen our muscles and bones. They might make our vision sharper. These mutations are beneficial mutations. Causes of mutations. Germ cell mutations are inherited across generations. Germ cells are sperm cells in males and egg cells in females. Some mutations are caused by mutagens in the environment. Sun's ultraviolet rays may cause gene mutations. Prolonged exposure to X-rays may also trigger mutations. Radioactive substances like uranium and thorium can cause mutations. Infectious agents such as the HPV virus have ability to initiate mutations. There is a category of mutations which is not dependent on inheritance or environmental factors. These mutations occur spontaneously during the interphase of cell life cycle when the DNA replicates. Gene mutation and chromosomal aberration. We are going to discuss different kinds of gene mutations in this video. Scientists use a different term, chromosomal aberration, for large-scale gene mutations, which alter the number of chromosomes or make structural changes to the chromosomes. When mutation is limited to a small number of nitrogenous bases, it is called gene mutation. Sometimes the word chromosomal mutation is used to refer to chromosomal aberrations. Alleles. The word allele is based on the Greek word allos, which means other. Alleles are different forms of the same gene. It is worth noting that genes are located in a specific region of a specific chromosome. Since we receive 2 copies of every chromosome, one from father and one from mother, there might be a slight difference in the genes in these 2 copies. These copies are called alleles. All alleles of a single gene represent a single trait. Dominant and recessive alleles. Since alleles are different forms of a single gene, there is a dominant-recessive relationship between these alleles. An allele of a gene is said to be dominant when it effectively overrules the other allele. The other allele is called the recessive allele. Let's understand it with an example. A growing child in mother's womb may get different forms of a gene that control eye color. In our example, the alleles received from mother code for the brown eyes, while those received from father code for blue eyes. The child's eyes would be brown if the mother's alleles are dominant and father's recessive. Eyes of the child would be blue if the father's alleles are dominant and mother's alleles are recessive. It can be of other color if the alleles received from both the parents are recessive and a dominant allele from either mother side or father side in family is received. Types of mutation based on cells involved. Let's come back to our topic, mutations. Based on the types of cells involved, there are 2 types of mutations, germline mutations and somatic mutations. Germline mutations occur in the germ cells or sex cells. These mutations may be transmitted to offsprings in which every cell in the offspring will have these mutations. Somatic mutations occur in cells other than the sex cells. Impact of the somatic cell mutations is limited as they cannot be passed on to the next generations. Look at the picture on your screen to understand the difference between the germline mutation and somatic mutations. Types of mutations based on scale. Based on scale, mutations can be categorized in two types: gene mutations and chromosomal mutations. Gene mutations are small-scale mutations or point mutations which are caused by changes in some nitrogenous bases of related genes. These mutations are restricted to the segment of a chromosome where the related gene is located. Gene mutations have several subtypes. Addition or insertion mutations and deletion mutations are potentially dangerous mutations. Substitution mutations are often less harmful. Chromosomal mutations, on the other hand, are called large mutations as they occur in the large portion of a chromosome, or sometimes they may involve the entire chromosome. Chromosomal mutations are also called chromosomal aberrations. Chromosomal mutations are of two types: structural mutations and numerical mutations. Structural mutations bring in structural changes to a chromosome. Duplication, inversion, deletion, and translocation mutations are some of the structural mutations. Numerical mutations are those chromosomal mutations that cause a change in the number of chromosomes. Trisomy 21 is an example of a numerical chromosomal mutation wherein there are 3 chromosomes instead of 2 at the position 21. It's time to learn different types of mutations in greater details. Let's first learn about some point mutations. Addition or insertion mutations. In addition or insertion mutation, a new nitrogenous base gets added to the gene carrying position of a chromosome. Let's understand how this seemingly trivial change can interrupt the protein synthesis process in the cell. Since proteins are extremely important for running our bodies, the impaired protein synthesis process may create havoc to our bodies. In our protein synthesis video, we have seen how our cells make protein on the basis of instructions written in genes. In the transcription process of protein synthesis, an mRNA molecule gets created on the basis of DNA sequence, and this mRNA molecule exits the nucleus and goes to ribosome to complete the translation process. On your screen, you can see a normal DNA sequence consisting of 12 nitrogenous bases AAA, ATG, CTT, and CTC. In this example, these bases are assumed to be the instructions for creating an essential protein. Please note that here A refers to the nitrogenous base adenine, T refers to thymine, G refers to guanine, and C refers to cytosine. During the transcription process, a small messenger RNA molecule is formed on the basis of this DNA sequence. Now this mRNA molecule leaves the nucleus and attaches to the ribosome for completing the translation process. In the translation process, the nitrogenous bases on mRNA molecule are read in units of 3 or triplets, which are called codons. For each codon triplet, there is a specific amino acid which is chosen by ribosome to form a peptide chain. A Codon-chart in the form of a wheel is shown on your screen, which has mRNA bases in the inner circles and corresponding amino acids on the circumference. Our mRNA molecule has bases UUU, UAC, GAA, and GAG. As for the Codon-chart, the bases UUU code for the amino acid phenylalanine. The second codon triplet UAC will attract the amino acid tyrosine. The third codon triplet GAA will bring in the amino acid glutamine. The fourth codon triplet GAG will also be bringing the same amino acid glutamine. All these 4 amino acids will create a peptide chain which eventually forms the requisite protein. This is the normal way of protein synthesis for the given DNA base sequences. Let's now see what happens when an addition or insertion mutation happens. In addition or insertion mutation, a new base C gets added or inserted at the 7th position in the DNA base sequence. This addition alters the DNA sequence into AAA, ATG, CCT, and TCT. Based on these altered DNA bases, the messenger RNA will be formed with the bases UUU, UAC, GGA, and AGA. The change bases in mRNA will impact the translation process, which will now create a peptide chain with amino acid phenylalanine, tyrosine, glycine, and arginine. You can notice that the first 2 amino acids are the same, but the third and the fourth amino acids have changed due to the mutation. A peptide chain with a different amino acids will create a different protein which may be undesirable or even harmful to the body. In our example, the insertion mutation happened at the 7th position of the gene. Had it happened at an earlier position, the protein synthesized would have had more incorrect amino acids. Deletion gene mutation. Deletion mutation occurs when one or more than one of the nitrogenous bases from a gene sequence gets deleted. Let's understand how such a small change may badly impact the protein synthesis process, which is crucial to our cells. Once again, you can see the normal DNA sequence of a protein synthesizing gene consisting of 12 nitrogenous bases. These bases are AAA, ATG, CTT and CTC. The deletion mutation deletes the fourth base A, causing the gene sequence change to AAA, TGC, TTC and TCC, assuming the DNA has base C after the sequence of bases belonging to this gene. The mRNA codon triplets created on the basis of the change gene would now be UUU, ACG, AAG and AGG. In the translation phase, the codons of mRNA would bring in 4 amino acid molecules, out of which only the first would be correct and the remaining 3 would be different from what they would have been had the deletion mutation not taken place. A peptide chain formed out of these amino acid molecules would not be able to make the correct protein essential to the cells. Gene mutation, substitution. In this mutation, one or more nitrogenous bases of a DNA sequence changes. If the changed bases belong to a gene related to protein synthesis, it impacts the protein making process, but the impact is limited. It only impacts those amino acids in the peptide chain which are brought by the changed codon triplets. The triplets before and after the changed bases remain unaltered, hence the substitution mutation is often a lesser evil than the insertion or deletion mutation. The insertion and deletion mutation has more serious consequences because they alter every codon triplet after the position where the mutation has taken place. That's the reason the insertion and deletion mutations are also called frameshift mutations. For the same reason, the cancers arising out of insertion or deletion mutations are extremely difficult to treat. I would like to mention here that, for simplicity, the gene sequence in the insertion, deletion and substitution examples have been kept short. In reality, a typical gene sequence is quite long. So far we have been talking about point mutations. Let's now briefly discuss some chromosomal mutations. Duplication chromosomal mutation. A duplication chromosomal mutation adds multiple copies of a single gene within a chromosome, hereby increasing the genetic material within a chromosome without increasing the total number of chromosomes. A rare genetic disorder, Pallister-Killian syndrome, is caused by duplication of the genetic material in chromosome 12. This disorder impacts brain development and impairs vision and hearing abilities. Deletion chromosomal mutation. A deletion chromosomal mutation occurs when a part of a chromosome detaches from it. Depending upon the size of the deleted portion, the mutation is less or more lethal. A moderate sized deletion mutation creates Williams syndrome, which causes a large forehead and reduced chin and nose. Inversion chromosomal mutation. In inversion chromosomal mutation, a portion of a chromosome detaches, gets upside down and then reattaches itself to the same point. Inversion mutation in chromosome 12 was the reason behind world's tallest teenager's unusual height. Translocation chromosomal mutation. When a portion of a chromosome detaches and gets attached to some other chromosome, it is called a translocation mutation. We will discuss chromosomal mutations in greater details in some other video. We have reached the final slide of this video and also the video series. The topics learnt in this series are going to provide you with essential background to comprehend the units 9.1, 9.2, 9.3 up to 9.8 of the Anthropology Paper 1. I hope you find this video to be helpful. Kindly put your comments and suggestions in the comment box. Thank you.